Thank you very much. And thank you very much for the invitation and all the support from the Gnatwork network. So the title of the project is Understanding Host Heterogeneity to Vector-Borne Disease Exposure and the Implications for Modeling Disease Transmission and Improving the Design of Controlled Interventions.
Do you have any slides there, Orin? Because we can't see them at the moment, so please share your screen if you are looking at some.
Okay. It says I can't share my content for some reason.
Shall I see if I can share it? Oh,
hang on.
No, it is coming up.
It's come up.
You're on.
Good.
It's up.
Sorry about that. Right. So the title I've just told you. So as a means of background, so transmission models of vector-borne diseases generally predict that heterogeneity and exposure to vector bites is a very important determinant of infection outcomes, both at the individual level and at the population level. And this influences the persistence of transmission under different interventions. Okay, now vector exposure patterns, both age and sex specific, and also the differences between individual biting rates, are used to guide epidemiological surveillance and to address control policy relevant questions. But the practical tools to measure exposure to vector bites, which usually relies on human landing catches, is both very labor intensive and considered unethical, particularly in high-risk groups that are often children. So therefore, the transmission models have relied until now mostly on infection data to infer the exposure patterns, but it's important to measure these things independently in terms of the outcome of the models. So what's the solution? Well, we know that host exposure to arthropod vector saliva during blood feeding induces a quantifiable Ig antibody response in the host, and that these antibody responses sort of accumulate and decay with discontinued and continued exposure, making them a good target and candidate biomarker of current exposure, and in some cases of infection itself with the pathogen. But for many vector-borne diseases, anti-saliva assays have not been developed, or they haven't been applied in an epidemiological sense. So the aims of this project were to develop and apply anti-saliva assays for blackflies, targeting the Damnosum sensulatu group, which is the main vector of onchocerciasis in Africa, and for sandfly Phlebotomus orientalis, which is the main vector of visceral leishmaniasis in East Africa. And with these assays, the idea was to quantify the heterogeneities in individual exposure to bites with and between foci or villages by measuring responses to the salivary gland homogenate or and recombinant antigens, particularly in the case of Phlebotomus orientalis assay, which the team developed just prior to this project. And with these data to develop individual-based transmission models, incorporating biting exposure data independent of infection data to explore the implications for the control of onchocerciasis in ODA countries or the visceral leishmaniasis. And along the way, we developed scientific protocols that you can see on the Gnatwork website, and we provided training to the young researchers in ODA countries, including in Ghana. We trained them in salivary gland homogenate preparation in the field, and in Sudan, we trained them in sampling designs and preparation of quantitative ELISA assays. In addition, we also trained non-ODA researchers, and you're going to hear from a couple of those people today. So the team comprised of myself at the University of Warwick, Professor Maria Gloria Bassanez at Imperial College, Peter Wolf in Charles University, Prague, Mike Oseyentenembwana in the Council for Scientific and Industrial Research in Ghana, and Dr. Maha Osman from the National Center of Research for Khartoum. Now, given the time limits, we don't have time to cover all aspects of the project today, so we're going to focus on the development of the novel blackfly assay, which is going to be presented by Laura Willian, and also using that data, the development of the mathematical models and the implications for control, which is going to be presented by Philip Milton. And just before I pass it on, I just have to say that the risky part of this project actually was developing this novel assay for blackflies. So we're very pleased that we have succeeded in doing that. So over to you, Laura. I think I'm still controlling the slides.
Yeah, thank you, Orin. So in order to measure exposure to blackfly bites, we developed an immunoassay that uses blackfly saliva. We use saliva because it's well known that vectors are not flying syringes. They are in fact flying pharmacologists that have anti-hemostatic, anti-inflammatory, and vasodilatory molecules present in their saliva. Also for sandflies and mosquitoes and tridamine bugs, we do know that antibody responses directed against saliva or salivary proteins, that these can actually be good markers of host exposure against the vector. But what about blackflies? There have been no human studies performed to use anti-saliva antibody responses as a marker of host exposure. The salivary transcriptome of only 3 blackfly species are known, and none of these is Simulium damnosum sensolatum. And we do know that their saliva is allergenic, but we didn't know if we could actually use it to measure and differentiate exposure levels. So to develop this immunoassay, you can move on to the next slide, Orin. We went to Orin.
Yeah, sorry, it's not moving. Hang on. There we are.
Yeah,
Sorry.
we went, that's fine. We went to the field in Ghana, and there we collected blackflies and blood samples. Blackflies were collected through human landing catches near the Prue River. This is located in the Bono East region in Ghana. You can see the region, it's encircled on the map of Ghana in your left lower corner. We collected blackflies for 15 consecutive days with a daily target of 200 flies. And afterwards, the flies were also molecularly identified to assure that they were all from the Simulium damnosum complex. The salivary glands of the flies were then dissected on the same day by anesthetizing the flies, by putting them in a -20-degree freezer. And this is also where the training took place. So all the co-workers that joined us in the field, they were actually trained in salivary gland dissection. And in total, we dissected 940 glands. Also, blood samples were collected at the same time in the same Bono East region. We sampled in 4 different communities. These were located quite close to each other, but had allegedly different blackfly biting rates. And then also blood samples were collected in the capital of Ghana, Accra. This is a blackfly-free region. So these samples were then used as negative control samples. In total, we collected 1,000 blood samples. And then using all this material, we developed a immunoassay in the format of an indirect ELISA. So that means that we use the salivary gland homogenate as a target. So it was coated on the well. And then a conjugated secondary anti-human IgG or IgM antibody was then used to measure the presence of IgG and IgM. As you can see in the graphs, we were successful in developing both the IgG and IgM immunoassay. So what is shown here are the distributions of the standardized optical densities for negative controls versus people living in the Bono East region. The left graph shows the distribution for the IgG assay, and there you can see a clear distinction between the negative controls and the residents of the onco-endemic area. The same is true for the IgM assay, which is shown on the right. Although the difference between the two looks smaller, it's still significant. The fact that the IgM assay overall shows lower responses is possibly due to its shorter half-life compared to IgG responses. And the results of the development of the assay have been submitted to PLOS NTDs. Now, in the next step, we then wanted to see if we could actually characterize the most immunogenic proteins present in blackfly saliva. And for this, we ran an SDS page in order to separate the proteins. And the gel was then stained by 2 different staining methods. You can see this on the left. So Comassi and a silver stain was performed. And then several bands were cut from the gel and subjected to mass spectrometry analysis. We also performed an immunoblot to see which of the proteins were most antigenic. For this, we included negative controls from ACRA, which is shown in the blue frame. And you can see they do not react at all with any of the proteins present, which is very good. And then the people living in the Bono East region, that's shown in the green frame, they did react with several bands with different intensities. Very interesting is also that the intensity actually decreased when the optical density as measured by ELISA decreased, which is not always seen. What is also shown is that the samples that were in fact negative according to the ELISA cutoff value, so these are the most right samples, they still reacted with some proteins. And this is very interesting, as these could actually be the most antigenic and probably the most interesting for future studies. Then the mass spec analyses identified that there is apirase, hyaluronidase, and antigen 5 related protein present in blackfly saliva. These are all well-known salivary proteins, but definitely as a next step, the sialate transcriptome of Simulium dominosum sensolatu should first be unraveled in order to really identify immunogenic salivary proteins and then recombinantly express them and use them as a marker of blackfly exposure. I will now give the floor to Phil, who will talk about the trends that are observed and the modeling done on these data.
Yeah, thank you, Laura. So what we really wanted to look at was how the titers vary between individuals. How does antibody vary between individuals? And so the first thing to do that sort of analysis is, of course, just a regression model. So we started off with a gamma GLM, a generalized linear model, with the inclusion of some specific covariates of age, sex, village, so the village in which the individuals live, as well as an age and sex interaction term. In general, what we got from the regression is the idea that with age, individuals' antibody titers tend to decline, and that males tend to have a lower antibody titer than their female counterparts. Overall, sort of the general trend of the dynamics here would be that sort of males tend to have a slightly higher optical density, so a higher titer at early ages, but it declines more rapidly. You can sort of see that in D on the panel, figure D on the panel on the right. With females maybe starting a little bit lower, but sort of not declining quite as rapidly. So we get sort of a general idea of the overall dynamics. In terms of the differences by village, things were of mixed significance, but there does appear to be somewhat of a difference between the villages that they sampled in terms of the optical density. Yeah, next slide, please. So then we want to move from just sort of statistical trends to dynamic trends. You know, what are the factors that are driving changes in antibody titer? Well, to do that, we looked at specifically focusing on the IgG data. We used something called an antibody acquisition model. These are modeled using something called ordinary differential equations, but essentially you can just consider it as a process by which your exposure to blackfly bites allows you to acquire antibodies. And we also assume that antibodies don't stay within individuals forever, they decay. So we have a rate at which antibodies flow out, and the balance of the rate at which you're being exposed and antibodies are coming in and the rate at which they're decaying allows you to sort of get an idea of what an individual's current antibody titer should be. And that creates sort of a very basic model up there. It's model 1, the exposure only model. We also wanted to sort of push that one step further. We wanted to assume that maybe with continued exposure to salivary antigen, that an individual's immune system may become somewhat desensitized to the process of acquisition. So we assume here that your cumulative past exposure to blackfly bites might also slowly result in you responding less and less to the antigen. That created sort of a second model that we call the desensitization model, which is below. Next slide, please. So when we fit these models to the data, both models are actually able to fit the data quite well. The sort of greeny data on the left is males, and the sort of reddy pink data on the right is females. And the two different lines, the dashed line is the desensitization model, so the one which includes sort of a reduction in acquisition with cumulative past exposure, while the solid line is your exposure only model. You can see that they both fit the model okay, the data okay, but the model that was best was actually a desensitization model. So we carried that model forward. So what's really, really interesting is that the patterns of exposure that we're deriving from these from this antibody data actually fits very nicely with what we normally see in onchocerciasis. So on the left, on the very left panel, you see the profiles of microfilariae. So this is the age and sex-based microfilarial loads we see in individuals. What's really great is that on the panel on the right, when we've taken the information we've got from this antibody assay, we're actually really able to capture a very similar profile. So in other words, from the antibody data, we're actually able to predict quite nicely what we see in the field in terms of individuals' parasite loads, which was a really nice outcome. Next slide, please. I think it's for MG. Is Maria Gloria there?
Yes.
Oh,
thank you, Phil. So I am going to give the conclusions slide. So as you were able to ascertain, our anti-salivary assay for Samilium damnosum was successfully developed and used to investigate exposure to blackfly bites in the onchocerciasis endemic areas of Ghana in which the fieldwork was conducted. These results were used to understand the age and sex specific patterns of antibody responses to blackfly bites, and also to develop alternative models that would be capable of explaining such patterns. As you heard from Phil, the best fit model was the model with desensitization. And that model helped validate our previous exposure assumptions. So our model EPIONCO-IBM does have age and sex specific exposure patterns, and it has been used in a full stochastic transmission model. But what was very interesting about this work is that the antibody work helped validate our previous assumptions that had been derived from, in this case, infection data rather than direct antibody data, but the two very much match. Why is this important? So why is understanding patterns of age and sex specific exposure so important? Well, we have implications for the burden of disease. So as you saw, if exposure is, say, greater earlier in life, that will have implications, for example, for the development of epilepsy, onchocerciasis-associated epilepsy later in life. And this very much depends on children's exposure and accumulation of infection. It has also treatment impact, because so far with ivermectin, which is the drug of choice to treat and control onchocerciasis, children under 5 are not currently treated. But as you saw, their exposure can be substantial. So they could play an important role in the maintenance of a reservoir of infection and transmission. It also has implications for serological monitoring. What age groups are the most informative for serological monitoring? Again, depending on exposure patterns, we will be able to better tailor serological monitoring. It also has implications on surveys for elimination. And finally, this has been done in Ghana. The data that we obtained from Ghana matched our assumptions for Cameroon, but can we look at spatial variation in exposure? How does this change from location to location? And would that then help us to better tailor transmission models to particular locations? And I think this is our last slide. So we would like to thank everybody in the Ghanaian team, Michael Seatueneboana, Francis Verrier, who has been mentioned in other presentations as well, Laura Willen, Phil Milton, our modeling teams at Imperial College, Orin for coordinating all of us and keeping us on track, and obviously the Gnatwork for fantastic support for this project. Thank you very much.